Morning, everybody. We're really lucky to be joined this morning by Professor Laura Piddock, who is our keynote speaker for today's meeting. Laura is a Professor of Microbiology at the Institute of Microbiology and Infection at the University of Birmingham, and her current research focuses on the understanding of mechanisms of antibiotic resistance as the basis for drug discovery, as well as multidrug and regulation of multidrug efflux pumps, and until recently, inhibition and transfer of plasmids between bacteria. Laura is currently GARDP's Director of Scientific Affairs, leading the Discovery and Exploratory Research Programme, as well as external scientific affairs, including Revive. Today, Laura is going to talk to us about the Global Antibiotic Research and Development Partnership, GARDP, filling gaps that others don't. Thank you very much. Over to you, Laura.
Thank you, Jonathan, and thank you, Matt. And first, I'm going to apologise to everyone that I can't show my slides myself because my computer decided to go into automatic updates, which we found out 5 minutes before we went into the meeting. So thank you for giving me this opportunity to talk with you today. I'm very keen that we share the work that we're doing, particularly on two programmes at GARDP. And GARDP is a not-for-profit drug developer. So next slide, please. So the vision of GARDP is that all infections are treatable for everyone, everywhere, and not just for high-income countries. Next slide, please. So our mission as a not-for-profit organisation is to bring together the public and private sectors to develop new treatments for bacterial infections. We are focused in our work, and our first focus is on bacteria that are within the WHO priority pathogen list. But our second focus is on infections, diseases in populations that are disproportionately affected by drug resistance. And then our third focus, we predominantly do late-stage clinical development and access of new treatments. Next slide, please. Just to make clear where we fit into drug discovery R&D, the black band across the top shows you the typical pipeline from discovery to preclinical to clinical development. And the blue band immediately beneath shows our involvement in these areas. And as I've just said, our majority activity is clinical development, but we've spent a lot of time this year, and we'll be spending more next year, on developing access activities, because there's no point in getting a new drug approved if you then can't make it accessible to people. But as shown on the left-hand side, we do have discovery and exploratory and preclinical activities, and I'm going to talk a little bit about those in a minute. So there are other players here, and just to show you at the bottom, as I'm sure you're all aware, that many small companies and large pharmaceutical companies work across the development pipeline. But in the middle are what we call other players, and these are people who have facilitated funding for antimicrobial drug discovery R&D. Next slide, please. So this really outlines the activities that we do in GARDP's R&D. So focusing first on the middle band of clinical and pharmaceutical development right the way through to implementation, we focus on 4 areas: serious bacterial infections, neonatal sepsis and paediatric antibiotics, and sexually transmitted infections. So we have a phase 3 trial on zoliflodacin, first-in-class drug developed by Entasis, and we work in partnership with them. We are the sponsor of the phase 3 trial, and that's ongoing at the moment as a new treatment for gonorrhea. We have another program, serious bacterial infections, where we are working with Venatorix to co-develop taniborbactam cefepime for the use of serious gram-negative bacterial infections. And we hope that should the adult trials be successful, we will go on and develop its use for children and babies. If you could just press forward, thank you. So I'm going to talk about our Discovery and Exploratory research, which is one of the teams that I lead. And then, next button, please. I'm also going to talk about the scientific affairs activities that we do, and predominantly our Revive programme, because I think both of these will be of interest to this audience today. And who knows, some of us may end up working together. Next slide, please. So firstly, our discovery activities. So we aim to, again, to fill critical gaps in the pipeline, but this time very early in the pipeline. So we want to build a portfolio of new chemical entities with activity against those bacteria that are in a critical or high priority WHO list, and specifically with activity for Klebsiella and or Acinetobacter. Another factor for us is that we don't want chemicals that are cross-resistant to other classes, so this is a no-go for us. And ultimately, we want to find new chemical entities that will proceed into our clinical serious bacterial infections programme. We work on our projects in collaboration with others and by developing consortia. So not only to allow us to identify new chemical matter, but to progress those prioritized hits into preclinical development and hopefully onward. But crucially, we focus on novelty and ensuring that we don't work on things that others are working on. We have a limited budget for this programme, and we want to use it really carefully and wisely. Next slide, please. So why are we interested in doing gram-negatives when others with far greater resources than us have not been so successful? So we can learn from that work, and GSK, AstraZeneca and Merck have all been very public about how they've screened millions and millions of compounds and ended up with none that they can proceed with activity against gram-negatives. So there's really two main reasons that they've been unsuccessful at the time, which was mostly in the 2000s. The screening strategy that they used and the chemical property of the libraries they were screening. So what do I mean by the strategy? Well, many of the screens were enzyme-based or cell-free. And although they got hits, some really good inhibitors, the medchem was unable to resolve and get activity against whole cells. And the challenges for gram-negative are greater. Even if you could get, or they could get activity against whole cells, this was typically against efflux deficient E. coli, such as a Tol-C mutant. And as soon as they went into wild-type, clinically relevant bacteria, or even just into wild-type E. coli, they couldn't get activity that was resolved by chemistry. And then finally, the libraries they were screening were not designed for antibacterials, they were designed for other areas, and they didn't have sufficient chemical diversity, or they were in an inappropriate chemical space. But we didn't know that at the time, these companies didn't know that at the time. This is what we've been able to learn from that research. Next slide, please. So what do I mean by chemical space? If you look in the top left-hand side, this is the data from a paper from 2008, and this is for approved drugs. And in pink, in that circle, is the chemical space where approved anti-gram-negative drugs resided at that time. And this is really building on work first done by Hiroshi Nikaido in the 1980s on beta-lactams. And he showed very clearly that the majority of effective drugs had a molecular weight of less than 600 Daltons, and they were hydrophilic. If you look below to the middle chart, you can see that the typical screening libraries had very little diversity. And whilst they may have been the appropriate molecular size that we associate with anti-gram-negative activity, they were frequently more hydrophilic than desirable. And then the third box below, you can see the diversity in antibacterial compounds published at that time from academic groups. High diversity, but again, somewhat limited within the desirable space for gram-negs. More recently, Paul Hergenrother has published data on the so-called entry rules, as you can see on the right. And adding to this, the compounds that accumulate well within bacterial cells, particularly E. coli as shown here, There's some flexibility in the molecule itself, and its three-dimensional structure is important. Next slide, please. So if we take here the typical activities done for discovery research, whether it's phenotype-based or target-based, where do we place the GARDP work? Next, please. So we've been fortunate in having libraries shared with us that have never been previously screened by companies who've exited the antibacterial research area but have continued making new chemical matter. Then we've done a lot of cheminformatics, some by the companies, to choose the chemicals within those libraries where they've got the properties that one can associate with optimal entry into and being retained within gram-negative bacteria. And we've been looking at some novel natural products as well. Next, please. So not only do we screen whole cells of bacteria, we screen highly drug-resistant bacteria. So we put the hurdle quite high for us so that we know that we'll be starting off with a limited number of compounds, and also, trying to find activity against these drug-resistant bacteria is going to be challenging. Next, please. And we also had a project, I'm using past tense here quite deliberately to illustrate to you that we're very focused in what we do and targeted. So we had a project on an underexploited target in gram-negative bacteria, and it was to synthesize new analogues of an old antibiotic that inhibited that target. And there was some work done in E. coli to show it worked in whole cells. Unfortunately, we became aware of a company that was working in exactly the same area, and so we closed the project down. We're very hard with our no-goes so that we spend our money carefully. Next slide, please. This is where we are with our screening. So we have a Japanese consortium comprising currently Takeda, Daiichi Sankyo and Eisai, who've made their libraries available to us. And we've also had a library, the Reframe library from Calibre. And what we've been doing with that is looking for potentiators of an antibiotic that's in clinical use, but that, as far as we're aware, no one is currently looking for potentiators to it. And we've been able to look at some open source libraries from Merck, MMV and DNDi, and none of these have been screened for antibacterial activity. So in the last about 20 months, we screened about 65,000 compounds. Of those, we've gone on and done 798 in a dose response curve. We've then done hit validation and a variety of cytotoxicity assays and ended up with 229. And we're currently doing hit expansion and profiling and a variety of other activities such as physchem, metabolic stability on 4 series. Some of the analogues the company has been able to provide to us, and others we have designed in collaboration with chemists at DNDi, and we're having those synthesized by a CRO. Next slide, please. We do other DR activities, discovery and exploratory research. Because we are now going into hit expansion and we hope hit to lead in 21, we now need to get clear quantitative criteria for our go, no-go decisions and what progresses through our cascade. For instance, we may not wish to continue working on all of those series, and we may just share that information with the community because others may wish to pick up on them. We're continuing to build models, and we're doing a lot of computational activities and then following these up with experimental approaches. So whilst the properties for molecules to get inside gram-negatives and stay inside, some of those we're fairly familiar with now, they're predominantly for E. coli, and we're trying to use our own research to make these somewhat more species specific, particularly for Klebsiella and Acinetobacter. And we are continuing to horizon scan for new and underexploited targets, particularly those with high resolution structural information, and where possible with at least one inhibitor that we could then look to do a collaborative project with. Next slide, please. We are recruiting, so if anybody is looking to move on, the postdoc is finishing next year, we have this new post in our discovery team, and full details are available on the website. Just to say, we're a flexible organisation, but our requirement is someone in Europe and would be able to, once we can travel again, come to Geneva at least once a month, as I do. Next slide, please. So I want to move quickly on to our educational outreach activities, particularly Revive. Next slide, please. So this is to advance collaboration and share knowledge. And we launched this because at the beginning of 2018, when I first joined GARDP, it was clear that many companies were exiting this space and their researchers were either retiring or going to work in other fields. And so Revive was something that was part of the strategy predating me joining, and we've been implementing this for the last few years. Next slide, please. So we have 3 goals for this: to facilitate learning, and we do this through webinars and being at conferences and hosting our own conferences. Connecting people, we have over 100 world-leading experts that you can work with should you wish to do so. And we share knowledge through antimicrobial viewpoints and an encyclopedia that we launched in late October. And there's a brand website for this. It's part of the GARDP portfolio, but we have a Revive website where all of these activities can be found. Next slide, please. So this is just an example of the conferences that we've been at, and symposia, workshops and boot camps. On the right-hand side, the ASM ESCMID boot camps that we would have done, we did as live webinars, and these were done in collaboration with Carbex, Wellcome Trust, Repair Fund and JPI AMR. And then highlighted in red here is our conference that will be the 2nd and 3rd of February next year. It is targeting the Discovery and Exploratory community, although there's a couple of presentations that are different. It's free, it's online, and we're doing this in collaboration with BSAC, and guest partner is the IRAD JPI AMR Natural Products Discovery Consortium. So I do hope some of you can join. Next slide, please. These are some of the webinars. I've just picked some out here on the left-hand side that might be of interest to the audience today. So if you wish to pick up some of the knowledge from some of the experts in areas that you're unfamiliar with, please do. We had our record number of attendees for the artificial intelligence one. We had a keynote by Jim Collins and other experts in the field. Nearly 800 people registered for that, and that was very successful. All freely available. You can look at these anytime. And on the right are 2 webinars that are open for registration now. And the one by Pat Bradford and Lisa Miller may be of particular interest to the audience today. Next slide, please. Viewpoints, a wide range of topics. Just want to bring to your attention the one on the 2 databases, Antibiotic DB and Spark. DB has lots of molecules in that were not developed, although some have been since, through the pipeline, and that was a project that Tony Maxwell worked with my team on. We continually update that still. And then Spark, which is led by Pew Trust, which aims to have molecules with properties for getting drugs in or staying inside cells. These are really useful resources. Again, I think of particular interest to our audience today. Next slide, please. Then our encyclopedia, just to show you what it looks like on the left-hand side, you can either search or you can scroll through the alphabetical list. If you click on any of those topics, it brings you to the screen shown on the right, and you can get a brief definition. If you need more information, click on the link. You can also click on the tags or go to the resource library, which is the back end of all of this. That's freely available too. And you can pick up webinars on these topics or viewpoints or conference materials from others. So please have a look at that. Do let us know if there's any terms that are missing and that you would like to see featured. We're adding 5 to 10 a month at the moment. For some of the terms, we already have videos by experts, so a short explanation, which is very helpful. Next slide, please. So that was a real whistle-stop tour through 2 of our programmes, and this slide just features some of the funders of GARDP, also shows our founding organisations, the WHO and Drugs for Neglected Diseases, and we've been an independent foundation now for 2 years, and I do hope some of you would like to work with us. Thank you.
Thank you very much, Laura, that was a really great talk, thanks.